So what I want to kind of summarize a little bit, I think we heard a lot today that the microbiome, there's, I think, a lot of new insights. I think we need to understand there are different approaches to the microbiome, health versus disease. But I think, as you can see, metabolic pathways, they always keep coming back, like bile acid, short-chain fatty acid, and so on. And ultimately, that really brings us back into how important are actually enteropathogens. Are they the important ones, or is the whole GI disease microbiome connection more important than focusing so much on those potential enteropathogens? So again, that's the opinion of myself, not Nestle Purina. I have a few disclosures. Probably the most important for this talk is that our laboratory at Texas A&M, we offer microbiome testing, including the dysbiosis index and some other enteropathogens. So I think it's important for me to highlight one thing. That's we need to revisit how we look at the GI tract. I think GI physiology really is different than what we learned when we were in vet school. And the reason for that is that we really, until recently, we didn't really understand the microbiome. And so when you look at the microbiome, this cartoon on the left, that's how the GI kind of looks like in a schematic scheme. So we have this microbiome here, and diverse, the more diverse, the probably better. But we also have this very important mucus layer here. This in yellow is this mucus layer. This protects, or not protects, but separates out whatever happens in the lumen of the GI tract from the epithelium. And if everything is in balance, then we probably have a very balanced immune system. There's a pro versus anti-inflammation. It's not one or the other, it's a balance and so on. And this is actually one of our PhD students, Chi, who did this really beautiful picture. This is the colon of a cat, a healthy cat. And when you think about, this is the lumen in the GI tract, that's what we usually see as feces. That's until now, we always considered more of a waste product. And you can see how many bacteria are present. There's a lot of bacteria, estimated at around 100 trillion cells. Then this mucus layer here that is really probably one of the most underrated safety features in the GI tract, because this mucus layer separates all of this, what happens in the lumen, from the epithelium. And then if everything is intact, that really means probably we have a healthy GI tract. And what's important to understand that it's not just one or the other, both of them are one unit. The gut microbiome is a part of our physiology. We know that now very well. There's some pathways that only microbes can do. But it's also important that microbiome is not isolated. We cannot really take it isolated. It's like the ecosystem in your backyard or wherever. You cannot really plant something if the rest of the environment doesn't fit. If the soil quality is bad, or if, for example, we don't have enough the bright water and so on. So it needs to be that we cannot change the microbiome if we don't really take into account. But the second component is also important is, if there's some change in the microbiome alone, meaning, for example, we induce dysbiosis by antibiotics, as long as all those safety features are here, I think the gut is very able to withstand a lot of changes. So the gut is very resilient. It makes also sense because over time, there's a lot of how to call it, damage to the gut performed and so on. So I think the gut needs to be resilient. So as long as this mucus layer, everything else is okay, what happens in here may be really buffered up. But if it's long-term, then we can imagine that this is going to be also like changed. But also importantly is, if this changes here, if there's inflammation, chronic atrophy, villous atrophy, and so on, the mucus layer is degraded, dysbiosis, then we really have one big component together. And it's not one or the other. Many different pathophysiologies happen at the same time, and they are very overlapping. And they're going to also very individualized. And that's why this chronic enteropathy is a very umbrella term. It's probably a huge gradient from animals that might be just on an abnormal diet, but the GI tract is normal, all the way to there's already damage in the GI tract, that we have dysbiosis, but mild enough to truly still be managed with diet, all the way to cell responsiveness. And as Linda today beautifully showed, it's so extreme that we have bile acid malabsorption in the ileum with dysbiosis together, and those are probably a reflection of the severity of those changes. But there's a huge overlap clinically, of course. So we know that the microbiome is an important organ. I think we could talk a lot about metabolomics and so on. I think that's the future, because the gut microbiome is a central organ system. That means when you imagine every single component that enters the GI tract and going to be metabolized by gut bacteria. But I think over time, if we focus on major pathways like short-chain fatty acid pathway, for example, the bile acid pathway, I think those pathways explain quite a lot of those changes. And again, it's an ecosystem. It is different from animal to animal, but generally the functions, they are very, very overlapping. So I don't think really, when you look at the major pathways, I think we can really make the next big steps in understanding how the microbiome fits into GI disease. So let me talk about a little bit of Clostridia. And I think the reason for that is that we still treat Clostridia a lot. There's still a lot of anthropathogen panels that are reporting, looking at Clostridia perfringens, Clostridium difficile, for example, here. But it was always difficult to say, are they truly the cause of the disease? And if they're the cause, do we even have to treat them and so on? So my talk is a little bit initially about Clostridium difficile. That's my main focus. And how does it actually play a role in GI disease? And then maybe a little bit summarize what Andy and Helene kind of their studies and how do they fit in the current literature and what are the next steps. So Clostridium difficile, nowadays renamed as Clostridioides difficile. There's a recent review article, and it's always the same. It's enigmatic. So it's always written in textbooks. There is an association with disease. It's a potential anthropathogen. But really, the studies are not very clear. There's an association in some studies, but not other ones. And importantly, many of those pathogens, when you try to induce disease experimentally, they don't really actually cause the disease. So there's an association there, but it's not really clear, are they the primary driver? So there's low prevalence rates in healthy dogs based on all those studies, a little bit higher in shelter and hospitals. Interestingly, some studies have shown that 58% of hospitalization dogs that visit, so those dogs that are visitation dogs that go to a human hospital as a companion animal, they often actually come back with Clostridium difficile. So there's thought that actually dogs get it from humans and not the other way around. In humans, Clostridium difficile is clearly associated with disease. Not everybody gets it, but there's a very strong association between the presence of Clostridium difficile and colitis. To diagnose it, you need actually both. You need the presence of the organism, so the antigen, and also confirm the presence of the toxin by different means. So there's no doubt about that there is this antibiotic-induced infection with Clostridium difficile. And there's also humans with inflammatory bowel disease, so chronic inflammation of the GI tract, chronic destruction of the GI tract. They also are quite often positive Clostridium difficile. And it actually makes sense once we come back to our results. There's also nice data shown that there is a strong association between bile acids and Clostridium difficile overgrowth. Okay? And the reason for that is that bile acids, when they normal concentration, so the microbiome converts primary bile acids into secondary bile acids. So normal secondary bile acids in a healthy gut prevents the germination of spores of Clostridium difficile. What that means is, if the microbiome is normal and C. diff actually is present, it cannot really germinate. So the normal microbiome controls those pathogens. But in humans, when you get antibiotics, so we go into a hospital, as a classical example, for an elective procedure, people get antibiotics that disrupts the gut microbiome, disrupts bile acid conversion. And ultimately, if C. diff is an environment, it can actually overgrow because primary bile acids, they ultimately promote the germination. So again, it's a very, very well characterized association between bile acid conversion and ultimately the germination of spores. And in humans with Clostridium difficile infection, FMT, so fecal transplantation, is really pretty much the best treatment options available. It's not the first options because of safety concerns, but those people who undergo 3 antibiotic trials, C. diff comes back, when they get an FMT, the success rate is somewhere up to 90% and so on. And one pathway is that this FMT restores normal microflora, which also ultimately associated with normal levels of secondary bile acids. There's also new pathways. There's also a new pathway that is suggested that it's still a strong correlation with bile acid conversion, but it's not necessarily the bile acid themselves do it, but the bacteria itself, the bacteria that are present there, they can produce and outcompete Clostridium difficile with other ones. So again, it's still the bile acid converting bacteria that are important, and there are 2 possible pathways. One of them is they are competing against nutrients, or they're converting bile acids. So still, we're talking about bile acid converting bacteria. What do we do now in dogs? In dogs, there is case reports. So I think there's clearly data out there that there are some cases that have been associated with Clostridium difficile infection, and ultimately when we treat them, they can get better. So there's this one case reports in an 8-month-old French bulldog with a 4-month history of intermittent large bowel diarrhea. In this case, the authors really confirmed both the genes by PCR, so the organism and the antigen. That's why ultimately there was an association. And then based on human studies or based on human data, they actually used an oral FMT, fecal transplants. And 2 to 3 days after this oral transplantation, the clinical signs improved, and the dog was also better negative for severe antigen. So there's definitely some cases out there. But there's also cases out there that we see Clostridia a lot, and it's not really associated with disease. So, for example, the question was about acute hemorrhagic diarrhea syndrome. This is a study already from 2015, where we looked at back then, we didn't understand the DEF toxin yet. Back then, the DEF toxin was not yet described. So we looked at toxins that are known, like the enterotoxin from perfringens, and ultimately Clostridium difficile. And very few dogs with AHDS were actually positive for the toxin, only 4%, which again, now looking at Helene's studies and Andy's study makes perfect sense, because very few of those dogs have abnormal bile acid metabolism, okay? But there's also studies out there that show that Clostridium difficile can be present in chronic diarrhea, or at least case series, but ultimately, we don't have to treat it, because in these dogs, there were 5 dogs that had chronic diarrhea. All of them were positive Clostridium difficile, and they were unresponsive to treatment with antibiotics. Yet, when animals with chronic diarrhea, presumptively chronic enteropathy, were treated with dietary chains, all 5 completely recovered. That again suggests that this organism may occur in dogs with other GI alterations, and maybe also as a side of dysbiosis with chronic diarrhea again. So this was a case series in the past. So what do we know again about the canine microbiome? How does it fit really with bile acids and so on? So we always talk about how complex and diverse the microbiome is, and that's really correct. And I guess when you look at your own ecosystem, when you look in your backyard, when you look everywhere, when you would start counting all the different plants that are out there, you would also find thousands probably, okay? But I think to really hone it down is functionally, when you think about it, you're still going to see most are the same. There's going to be specific trees, specific shrubs, and so on. So there's a so-called a core microbiome, likely. And this core microbiome, what that means is those are probably those that are present in every single healthy animal, and all the other ones, which may or may not be important, are just simply random. And you see this in the microbiome too. So this is a study that we recently are doing, where we look at the bacteria in healthy dogs. And we have 12 healthy dogs here. And green just indicates bacteria present, red, they're absent. And you can see nicely that actually not so many bacteria that are really present across every single healthy animal. And when you really count in only 25 species, we identified a total of 1,000 bacteria across all the samples, but only 25 were truly present in all 12 dogs, and then 36 in none of 12. And when we look at which are actually the most abundant ones, it is bacteria that we actually discovered already in the past. We did studies in healthy disease, and those are the ones that are always statistically came up. So there's a reason, and the reason is because those are major abundant groups. So Clostridium halononis again, Ballata faecalibacterium. So again, there is probably a functional redundancy in the gut, and some species are more common in healthy dogs, and they are probably the ones that are more functional. And so that's why over time, we were able to discover that there are some bacteria that seems to be more in healthy animals, more common. They're normal in healthy animals, and they go down in disease. And those are like Faecalibacterium, Turicibacter, Clostridium halononis. Those are bacteria that have specific functions. And usually in dysbiosis itself, they actually are decreased. And then with time, we can also identify bacteria that usually increase in dysbiosis. Some of them may be directly harmful, like E. coli. Some of them, like Streptococcus, may be just an overgrowth bacterium, okay? But ultimately, over time, over many studies, and somebody asked the question, is the index validated? I think now we have almost like 130 papers where we use this in different clinical studies, and it's a very reproducible kind of pattern. And what that means is that we can actually have reference intervals for those bacteria. But together, we can also, so we can say, is this bacterial group normal, abnormal? In some of them is a huge range, some of them normal range. But importantly, we can actually mathematically put them into one single number. So this one number, the so-called dysbiosis index, is a mathematical model that puts those together. And if the more those group decrease, the more those group increase, the more increase in dysbiosis we're going to have. And I think what we're discovering now more and more is that we can identify to some degree what a huge range of normal is. And the question comes up, are there differences in normal? Probably, like in any other organ system, any other markers. There are going to be small differences in this huge normal range. But importantly is that the higher the index gets, the more we know it's abnormal. And it's quite specific. Above 2, it's mostly we can be very specific as a major shifted microbiome. And even more importantly, we saw, like Linda said, the more higher the dysbiosis index is, those are the more dogs that seem to have this changes long-term and so on. And some of those, like the dogs with bile acid diarrhea, again, it's an overlapping feature, but generally means there is something major wrong in the GI tract. The higher the index, the more abnormal the microbiome. And our current theory is that there must be much more changes on the mucosal level, something in the GI tract that really makes this abnormal. And from our previous studies, we know that if you're in this area, that it's almost many of those animals are gonna stay persistently abnormal and so on. So I think that's important, where we can at least, to some degree, differentiate, is the microbiome clearly abnormal? And then we have this huge range of normal. That's kind of important. And this Clostridium hydronolis is really a fascinating bacterium. So yes, in dogs and cats so far, it seems to be the major bile acid converter. I think we have a kind of a 90 to 95% classification between associated with the presence of hydronolis and the absence. And what's fascinating to me, it's all over the world. It doesn't make a difference which part of the world we're looking at. This reference is very, very narrow. So most healthy dogs are really in this reference interval. And that's when clinically healthy dog, really just a very small subset, somewhere between 5 to 10%. In this study, probably even lower, depends on the pet population. But as you can see, they are decreased. But we also note that they're clearly abnormal. And the reason is because those dogs, when we measure the bile acids, they are not normal. That means they do not have bile acid conversion. And that by definition cannot be explained physiologically. There is a reason. So those dogs are clearly, that have decreased hydronolis, are clearly have already abnormal gut microbiome. This bile acids metabolism is really fascinating because simply the presence of Clostridium hydronolis, if it's there or not there, so when we measure Clostridium hydronolis by qPCR, you saw the reference interval. If it's decreased, you can really cluster the guts bacteria, even based on sequencing, total microbiome analysis. So just simply the presence of this bacterium is strongly associated with a shift in the gut microbiome. Again, most likely because of those metabolic pathways and so on. And we can see this as beautiful in the dysbiosis index. That means if you don't have hydronolis, as you can see here, there's a clustering in sequencing data, but there's also clustering that clearly associated with increased dysbiosis index. So hydronolis itself, it's important to also understand there could be some dogs that are low, have an index below 0, but they don't have hydronolis. That is already a mild dysbiosis. But the higher the index by itself, that means there's other bacteria that are also abnormal. And that really would highly suggest that there are major changes in the environment of the gut that really drives the dysbiosis. Dysbiosis cannot really, as such severe one, cannot be by itself. And there's really two explanations for what we're going to have. So I think it's important to understand that there's a huge difference if the microbiome, the GI tract is normal, the dysbiosis, the microbiome is normal, or there's maybe just a mild dysbiosis, maybe just a little bit of diets and so on. But generally, it's still quite diverse. So in a healthy gut, we have a normal gut microbiome, mucus layer, everything is normal. In a diseased gut, once this dysbiosis, at least the best we have at the moment, develops, it is probably already a marker that there's something major wrong on the GI tract, except if we exclude antibiotics. I'm going to come back to this in 1 second. But ultimately, what it means is that this chronic GI disease has been there for a long time. There are chronic destructions. We can see this, like Linda showed nicely, the dogs with chronic GI disease, for example, they have villus shortening of villi. They have those receptors, transporters, absorption function in it. So ultimately, we have inflammation in the GI tract, but we also have a functional dysfunction of the GI tract. The villus are shorter. That means we have less absorptive capacity and so on. And ultimately, when you think about it, what happens if there's food in the lumen of the GI tract there? Bacteria can drive over that. So it's a combination. It's inflammation. It is a combination of malabsorption, a local malabsorption. It's a degraded mucus layer. And it's really probably all of this happens to some degree, but in every individual patient, the extent is different. And I think that's why we have this huge range what the chronic enteropathy is. It's not just one different diseases. It's probably just a gradient of severity of those changes. And they are very difficult to predict in individual patients. But of all means, if we see dysbiosis, that already would indicate something major happened already. If we see mild dysbiosis, that we probably could assume distal. Again, it's not a perfect marker, but the concept is important because it also tells us there's a different way how we actually approach those patients. And we see this really, I mean, Andy and Helene have nicely shown that most dogs with acute diarrhea, even if they have severe lesions, acute diarrhea, they have very mild increase. And that's transient and most dogs become normal again. But if you look at dogs with chronic GI disease, and they could be all, there's no distinction. There's not a predictor of the therapy, but they could be food responsive. Those dogs usually have increased dysbiosis index, and many of them have a lack of Clostridium hydronolis, okay? And those dogs, again, as said, if we don't, usually they stay quite long abnormal. Even if they go in clinical remission, Those takes a while, over months to years, if they normalize, and some actually don't normalize ever. That again, the only suggestion would be there's something major going on inside the lumen of the GI tract. So there's a huge difference with the chronic diarrhea, chronic GI disease, not even GI and diarrhea. Diarrhea is not dysbiosis. Dogs don't even need to have diarrhea. Diarrhea is a different concept, but there's some changes, chronic enteropathy versus acute. And we see this nicely. What Helene showed from her abstract is that in the ants, the dysbiosis index is a little bit is on this higher one. We saw they have increasing Clostridium perfringens. Okay, there is a less slight dysbiosis, it's a mild dysbiosis, so the index is normal, but there's some groups out of reference interval. But when you can look at the symbiotic group, for example, as you can see, day 67 even decreasing more than the normal ones. But if you use antibiotics, I'm going to come back to this because that's important, is when we use actually antibiotics, we make it actually worse. So those are dogs with acute diarrhea at baseline. They have a very mild dysbiosis that ultimately increase on metronidazole, and some dogs actually stay on dysbiotic in long term and so on. And that's potentially a risk factor for the future. And the same with Clostridia melanonis. We can see most dogs with acute diarrhea are in the normal range, just barely below a little bit. Those that are symbiotic group, they actually stay in the reference interval. If we give antibiotics, specifically metronidazole, we're actually making this dysbiosis worse, and this bile acid conversion is important. And so we need to understand that all those changes depending on what it is. So we know that antibiotics is a big one. So we're going to come back to this one. Antibiotics can induce dysbiosis. But we also understand now that this association between changes in the gut microbiome and some things. And there's also another stage in puppies, for example. In puppies or in humans too, when we are young, when we are just born, we don't have yet an established GI tract. That means we don't have an established flora. And that's going to develop when we, dietary changes, then the gut's physiology adapts, and then also the microflora is going to normalize because we have this anaerobic flora. So in the beginning, in the very early stages, we have a lot of E. coli, we have a lot of environmental bacteria. And you can see, so those are completely healthy puppies. It's to expect that they have E. coli. And then you can see 5 to 6 weeks of age, 7 to 9, the E. coli decreases and goes into the reference interval of healthy dogs. And that's why also Faecalibacterium, for example, this important anaerobic group is low or undetectable in some dogs at baseline, but very quickly, 5 to 6 weeks, it actually normalizes. And so in puppies, we see that the first 3 to 4 months of age, they have a physiological dysbiosis. And again, it's normal for them because they have a lot of environmental bacteria, because the gut is not normal. But once they come physiologically, we can beautifully see that how those puppies at latest, I think 10 to 16 weeks, most of the instead of one is in the normal range and so on. And over 52, all of them span the normal range nicely again. But why is this important? Because when we come back now to this association between bacterial overgrowth and normal microbiome, is what we found in the study was very interesting is that we found in those completely normal puppies, in the first week of age, they had a lot of abundance of Clostridium difficile. But they didn't have diarrhea. There was no diarrhea and so on. And as you can see, suddenly week 7 to 9, we have this huge drop in Clostridium difficile without doing anything. But why is that? Remember the previous slides, because the normal flora develops in this time frame. And we have Clostridium melanonis. Clostridium melanonis is the bile acid converter. As you can see, it was low in most dogs at baseline, but at 7 to 9 weeks, it's pretty much present in almost every single dog. And we know that Clostridium melanosis converts bile acids, and you can see nicely, before we don't have secondary bile acid, just primary. And ultimately, we can see secondary bile acid conversion, and we notice this association within. So again, it's a beautiful example that when we have a dysbiosis, and in most cases, dysbiosis involves lack of Clostridium melanonis, a significant dysbiosis, we don't have bile acid conversion, and that's why it allows some of those bacteria to overgrow without necessarily causing the disease, okay? And so this was kind of the segue to a study that we just did. Melanie Werner she's now in Zurich, and we looked at some data that we have, where we could go back to samples that we know where we have dysbiosis index with Clostridium melanonis and also concurrent Clostridium difficile results. And so we looked at 358 samples. We also looked at 33 of those dogs. We had a very good described history. So what was the treatment response? And in a subset of samples, we also had results for ELISAs available. Was the protein present? And ultimately, what we saw that from those 258 samples, we found actually a quite high percentage of dogs that were positive Clostridium difficile on PCR, almost 36%, so very high percentage. But what was also very striking, and now not surprising anymore, is that there was a very strong association between the presence of dysbiosis, and the presence of Clostridium difficile. So 90% or 89% of dogs that were positive Clostridium difficile also had an increased dysbiosis index. And almost the same percentage also had reduced Clostridia nonis, which again, most of them we know not going to have bile acid conversion. So the pathway, the mechanism is pretty clear. So again, the odds ratio of carrying Clostridium difficile was almost 9.5 higher when the dysbiosis was high. Also importantly is, we said before, in humans and other animal species, Clostridium difficile is well established to be a pathogen, but in dogs and cats, it's not really clear. And this will also confirm it a little bit that only 0 dogs from the 16 actually were positive on the ELISA pytoxin. So it was very, very almost not present in our dogs. But again, most importantly is though, yes, we see a lot of dogs are present, positive Clostridium difficile. They have dysbiosis. But if you look at the treatment outcome, that it didn't really make a huge difference if they're negative or positive. The treatment outcome was the same. So in the end, most dogs in both groups were food responsive. A small percent was steroid responsive and not responsive. And again, it probably comes back to those in chronic GI disease. We have often the occurrence of dysbiosis. We often have a lack of Clostridium nonis. And because of that, ultimately, if Clostridium is present, it's not going to present every single animal, it allows it to overgrow. It also would explain why in the dogs with AHDS, the occurrence of Clostridium is very low, because as Andy showed nicely, very, very few dogs had actually lack of Clostridium nonis at baseline. They have a very different dysbiosis pattern, if that makes dysbiosis, but they're very mild dysbiosis because it normalizes pretty quickly. So ultimately, same thing when we go back now to clinically healthy dogs, and that's why it's always important to look at that sometimes, is that when we go back, when we look at the clinically 116 clinically healthy pet dogs now, we saw again the vast majority is in the normal range. But we found a few dogs that were actually dysbiotic, so they're clinically healthy. We also know now that dysbiosis can be present subclinically, again, could be either antibiotic induced or could be potentially an already indicated some suboptimalis in the GI tract. And interestingly is those same dogs that are here in red were also the ones that lacked Clostridium nonis, as you can see, they're the low one. And interestingly, 6% of those positive dogs tested healthy dogs for Clostridium difficile, and all of them had a reduced Clostridium nonis. And again, when you look at the study first in the beginning, where they looked at reference intervals of present healthy dogs that will actually match it. So we know kind of really strongly can associate also Clostridium difficile in subclinical dysbiosis. Doesn't have to be always clinical. But again, we know that not having Clostridium nonis is not normal. There's no real physiological explanation for that. So they definitely have a dysbiosis. The question is it already an indicator of an underlying GI disease process, or is it some remnant of antibiotic use? Okay. And we're doing studies now, like in humans, to see actually, can we get rid of Clostridium difficile? Just a proof of a concept. Again, probably not really necessarily because it's not the problem, but it just shows you again very nicely the correlation is that there's a dog that has been here, 3 samples, pre-FMT, was always positive Clostridium difficile, didn't also have Clostridium nonis, and after 1 FMT, we did 4 grams per kilogram mixed, and we can see that really 9 days later, 18 days later, Clostridium difficile gets pushed out, and it really correlates again because after FMT, we can really, it was not here, and we can really 4 days later, we can see Clostridium nonis comes back in the normal range. And we also measured the bile acids, and it also correlates it. So it's a very strong association with the presence of this organism and Clostridium nonis. So again, to summarize this part is that up to 90% of dogs are positive Clostridium difficile. They have a dysbiosis. It's clearly associated with Clostridium nonis, reduced liquid bile acids. In dogs, in cats, probably the same. We're doing a study now. The toxin is rarely found. And again, it is there, it is probably there, but it's not associated with clinical specific response. So in very, very few cases, now there could be some cases where it would be, but most cases going to respond to standard treatment with dietary approaches and follow up with chronic enteropathy. So they would strongly suggest that in chronic GI disease, where we know we have this dysbiosis, that if it's there, it occurs secondary to this dysbiosis. And again, the focus should be on the treatment of the primary underlying disease. Very briefly about Clostridium perfringens, we already heard about this a lot, is that, again, I don't summarize it, we know there are different toxins. In dogs and cats, it's mostly the type A. That means every Clostridium perfringens in dogs and cats has toxin A. Some of them might have enterotoxin. And again, we see this same thing in dogs, like Helene's study and also also Andy and Helene's presentation was that at baseline in acute diarrhea and acute diarrhea, we see this increase at baseline. But we also saw that it's self-limiting. It goes away with specific treatment. And that makes also sense when you go back now to studies nowadays that were in the past only had one time point. Yes, we found those associations. They were higher. That's why in the past we always thought there must be an association. But in the end, as you can see, even this study concluded, yes, it's higher significantly, but the presence of toxins was not associated with clinical parameters and cannot be used to predict disease outcomes. So again, those studies in the past now make sense with the new studies where we can follow them up over time. It's self-limiting, it goes away. It may be, in some cases, a trigger, probably in most cases or not, but regardless, it does not require any specific treatment. And again, Helena showed this one. In this study, again, they showed nicely that there was no difference if you give antibiotics, not antibiotics. Also, Clostridium perfringens itself was higher, but you can see also beautifully it decreases. So again, it's a self-limiting organism that is associated with some overgrowth, but we don't need any specific requirement for this. So the question is always, well, does it hurt when you give antibiotics? And I think in most cases, initially we don't probably, but there's really nice data out there that really clearly shows there is an association with giving antibiotics and dysbiosis. So this is a study we did together with LSU, Dr. Gashan's group, where we give first, in healthy dogs, we gave, they were also, as you can see, all of those dogs were in a normal interval from the dysbiosis index. We give them high-risk diet. And you can see, yes, diet changes the microbiome slightly. And it's makes sense. Diet can have an influence, but again, here we're looking at the total normal of all the dogs, or a huge proportion out there. And the index really indicates a severe shift. So there are minor changes in here. They may or may not be important, but if you really compare those changes here compared to metronidazole, you can clearly appreciate how severe the dysbiosis is that we get with metronidazole. And we stop metronidazole 2 weeks later, 4 weeks later, still, they go, most animals normalize, but there's still a delay in some animals. Most of them going to come back eventually, but it takes longer for some of them. And you can see this also in Clostridium melanonis. Again, all the animals within the reference interval, which is still fascinating how it's actually really stable, even any dietary changes we do. And then on metronidazole, it decreases here. And again, most animals normalize, but again, 4 weeks later, you still have a few dogs that do not have this bile conversion again. So they have a significant change in their microbiome that is long lasting. And more importantly, actually, when you start thinking about it's just a dysbiosis, but we also note that there's a lot of other things going on. And then in this paper, we also looked at other metabolites. For example, we looked at fecal lactate, so the whole lactate in the intestinal tract. And when you think about how little lactate is present compared to at baseline, you can see there was really no change when we had a dietary change. But if you compare to metronidazole, there was a huge increase. And again, I think in baseline it was like 5 millimolar was normal. Some dogs go all the way up to 150 and so on. So there's a lot of lactate in there, a lot of lactate buildup. It's an example that every single time with this huge gut microbiome, there are metabolic changes, and ultimately they probably cause some changes, maybe small ones, but you can imagine maybe repeated changes that could really affect the gut microbiome, the GI tract itself, the mucus layer, and so on. So there's definitely huge changes that occur on antibiotics, and they're probably not the best ones. And that would actually also be in line why in human medicine there are clear data out there where we know that antibiotic usage is associated with increased risk for some of those diseases that we see, some of those diseases in the GI tract, asthma, and so on. And I want to highlight something important. It's a small risk factor, and it makes perfect sense. It cannot be a big risk factor. I mean, the gut is a very robust system. It must be robust. It gets a lot of exposure to a lot of different, not toxins, but different things. But it's clearly a risk factor. And again, it brings it to the same principle again. If antibiotics are needed, absolutely. But I think in diseases that we see now, like acute diarrhea, even AHDS in most milder cases, if they're not needed, then I think it makes perfect sense to avoid antibiotics whenever we can. Because again, every dose that they get or repeated antibiotics, especially in young animals, definitely has this little risk factor and so on. So we have to always weigh this. So ultimately, perfringens, I think it's important to understand it is a common normal flora. Clostridium toxin A, for example, is present in everything. So most animals are going to have perfringens. Yes, there's an association with at baseline with an increase above the reference interval, but I think there's plenty of studies now that have shown repeatedly it's self-limiting and does not require any specific treatment for perfringens itself. So there's no primary treatment indicated, it's supportive treatment. The last few slides just about NetF, because I think that's also very interesting and important and actually takes a little bit of a different pathway, because I think NetF itself is probably very clearly associated with induction of lesions. So this has been just identified a few years ago, like 2015. It's a pore-forming toxin. It is a sample certain channels. It has been seen as Clostridia in AHDS. Those dogs have these severe lesions. And again, Andy and a report about permeability, they're severe, but surprisingly still only a small subset of them have really more severe permeability changes and so on. But we note that they usually adhere to the mucosa here. But interestingly is that there are studies out there that have shown there's a strong association. We saw the summary that 66% of dogs with AHDS were positive in Andy and Helene's study. But when we looked at a huge population of animals, we really found association only with acute hemorrhagic diarrhea syndrome. Okay, so in our own studies, we found that 45, so it's somewhere between 45 or 65%, 66% are positive in AHDS. But we didn't find any so far dogs, or very, very few in non-hemorrhagic diarrhea. No dogs were positive in chronic GI disease. There was one in exocrine insufficiency, and 0 in healthy dogs. Again, so what that would suggest means that, yes, they can be sporadically in healthy dogs, but generally it's not normal flora. So perfringens that carry the gene for those native toxins does not appear to be normal flora. But if they are in a dog, there is a high risk of developing the disease. I'm going to come back to this one. So again, we saw 66% in the study were positive for net death. But you can see it's also very self-limiting. So it goes away without any specific treatment. So day 1, day 7, day 21. And we also saw the study from Ziese, FIM+1 published, also the same thing. There was also, I think, back to close to 60%. And again, day 3, day 7, we see it self-limiting. So what does that mean, really? It's actually very interesting. I think the theory could be it's not normal flora. That means it's not normal flora because we don't find it usually in other phenotypes, not in healthy, not in chronic, not even in acute diarrhea that is not non-hemorrhagic. So it means maybe a lot of healthy dogs can just pick it up, but if everything is normal, it probably passes through because it's not going to cause disease in every single animal because it's not infectious by itself. And I think one important thing is that we need to also understand, like we talked with some difficile and bile acid conversion, things like that. There's always something second has to happen until you can have the disease. The gut is very, very redundant, very, very stable. And a good example is probably in chickens, whoever is familiar with necrotic enteritis in chickens. It's a very similar toxin. It's also called perfringens. It's the NET-B toxin, also a very similar pathway. And in chickens, it's a very clear, there's no discussion that this is the cause that causes necrotic enteritis, but we cannot induce it experimentally if nothing else is going on. So if you just try to inject it, nothing's going to happen. But if there's damage in the GI tract, most likely to Eimeria, so you can see that Eimeria probably destroys a little bit this mucus layer or like the epithelial cells, and then the toxin is there, and then maybe it can easier just and then cause the disease. So again, always showing that it's not always has to be there. We don't have to treat it because as long as everything else is normal in the GI tract. So to summarize up, I think is to understand is the eyes are probably a huge difference if the microbiome is normal. The gut is normal. The microbiome is in this huge range of normal. But we can clearly also understand that if the gut is abnormal, we can definitely with high specificity say this is the microbiome is abnormal. And that's often consequence either of changes, of those changes in the GI tract. It's not just inflammation. It's a combination of inflammation, combination of maldigestion, malabsorption. So inflammation can change the microbiome. Too much undigested foods, some bacteria can overgrow it and so on, this mucus layer, or antibiotic uses. But again, because if you have those changes often, then we can see some of those overgrowth bacteria. In some cases, yes, they can cause disease, but often they can be just there without any problem and so on. But especially when you talk about Clostridium difficile, Clostridium perfringens, E. coli, we know they're kind of regulated to some degree via the normal microbiome. So if two ends, I have a lot of collaborators, a lot of studies that we collaborate with, and those are really a great source of samples that we never could really see this perspective between healthy disease if we would not really have a lot of studies. And of course, I thank you so many companies and other granting agencies for their financial supports. And again, the information about dysbiosis and so on. But I think what we need to really keep in mind, this is the new GI physiology. It's not one or the other. It's a combination thing. And one other thing that I just want to maybe highlight is if something is once here, this is really a jungle out there, it's probably very, very or impossible to ever get rid of it. So we also have to think about it. There's going to be some potential enteropathogens in healthy animals that just are there. They're protected, they are things it, but it's not unexpected we're going to find it because it's almost impossible to get rid of them again. Okay. So with this, I'm going to end my talk.